Good morning. I'd like to say thank you so much for the opportunity to just share with you some of the research of the Macadamia Protection Programme, of course at FABI, the University of Pretoria. So if you think about the program, the focus is on integrated pest management. So if we think about IPM, that would be reducing the impact of pests and diseases by applying multiple different strategies. But successful implementation of these strategies are really fundamentally based on the knowledge of the species, understanding the origin and diversity of the species, understanding population dynamics, seasonal cycles of insects, as well as life cycle and epidemiology. So current research projects or focus areas are then either based on filling some of these knowledge gaps or are already working towards the development of alternative control strategies. But research also needs to be done in collaboration with continued monitoring of pests and diseases. So our contribution towards that is, of course, the FABI Disease Diagnostic Clinic. And so this is then, in actual fact, a service that we provide to the industry. So yes, it's about monitoring what we know. It's ensuring the identification of new pests and diseases. And it's about ultimately using the information that we obtain over time to help direct future or new disease or pest-specific focus areas. So if I look at an example, thinking of a disease, of course, Macadamia dry flower that is caused by new pest Lathiopsis macadamia was identified and confirmed to be present in South Africa via the Disease Diagnostic Clinic. If we look at the number of samples that we've received during the last 2 years, we need to acknowledge that this is of growing importance. Perhaps if we think about a pest, so one of the really good and well-known examples is that of the polyphagous shuttle borer. What I'm including here on the slide is some of the reproductive hosts from which we have observed really high levels of infestation. I also include pecan. So currently pecan is the only agricultural important crop from which we have really reported an infestation level infection rate. What about macadamia? Of course, it was first reported in South Africa in February 2019. Where are we at the moment? We know of 2 additional reports, that is all single trees from different farms in KwaZulu-Natal. But we should not only focus on the polyphagous shuttle borer, we also need to consider a symbionts that these beetles carry. One, for example, is Fusarium eulacea, and it's the causal agent of Fusarium dieback. So a very important pathogen and causing death of a large number of tree species. We know that the beetle feeds on it, but we also know that the beetle actually relies heavily on this fungus and needs to complete its life cycle. So for us in macadamia, the potential impact of Fusarium eulacea, so thinking about Fusarium dieback, was unknown. And so this was then the project of D. So what we did is we first of all conducted pathogenicity assays. It was also a cultivar susceptibility assay. So we included 9 cultivars. We did 2 separate trials, first of all on young trees, but we also did a detached branch analysis or an assay. So what the detached branch assay is all about is sort of drilling a small hole into a branch, inserting an overgrown toothpick, so overgrown with Fusarium eulacea. So in actual fact, we're trying to mimic the exact way in which the fungus will enter in the tree. So what about our results? And this is across the board. When we look at either the small seedlings that was used or the detached branch, all the cultivars and repeats, we only ever observed very small lesions. So this is actually really good news for growers. So what we report is we see that the Fusarium grows really slowly. It clearly lacks aggressiveness within the macadamia tissue. So perhaps the anticipated fear of Fusarium dieback, specifically on macadamia, is then less than what we initially anticipated. What we still don't understand perhaps would be the beetle being attracted to macadamia. So what is the probability of this beetle to start a mass attack? And so continued monitoring is required. For now, practical management on farms and perhaps specifically within the KwaZulu-Natal area, is to monitor these reproductive hosts that are in close proximity to orchards. So similar also to what Skalk talked about on Tuesday, is we are aware of quite a number of other beetles, so in other words, not Polyphagus shuttleborer, that we are reporting in our orchards. But these beetles we now are referred to as secondary, so they will only attack stress or dying trees. And so in this instance, I think managing or an identification of your tree stress-inducing factors becomes really important. So if we think about stress, we also then need to look at the research of Angel, and so she specifically works on Botrytis dieback. She's done a number of surveys, including all the main growing regions of South Africa, and for her survey, we specifically sampled from both diseased as well as healthy tissues. So dieback trees, but also really seemingly healthy orchards. Why would this be relevant? So we know that these species or species within the Botrytis are typically regarded as an endophyte. An endophyte is then a fungus living in healthy tissue without causing disease. But we know that many of these species can actually switch to a pathogenic state. So it's referred to as being a latent pathogen, and this typically when the host is under stress. So a very easy and short summary of her research, we obviously was able to detect sort of similar species, both diseased as well as healthy. But when we started looking at our pathogenicity assays, what I'm now highlighting is actually those species with the ability to switch to being a pathogen. So it's important to note that there are some species that up to this date, we've only ever identified from healthy tissues, but can actually become a problem. And so when we think of stress, this is some really exciting ongoing research in Australia where they're currently looking at the association of Botrytis dieback in relation to drought and also insect damage. And I'm hopeful that we will be able to share some of their data with you shortly. So of course, we were fortunate enough to visit them just before lockdown, and that was thanks to the Australian African University networks. But what is our ongoing focus? And in that small, Angel is in the process of building a picture for us to understand the movement of Botrytis in between orchards and even from the surrounding environment. So I'm just showing a brilliant review paper. And so this information, for example, if you think of other nuts as olives, pistachio, walnuts, is already available. And we are building this for macadamia. What I want to highlight is that in some of these other nut crops, we need to understand that flowers and nuts can also sometimes be affected by species within the Botrytis. And so again, these are just preliminary results where Angel specifically focused on diseased flowers, diseased nuts, and on what I'm highlighting here in blue are actually the species that we isolated. And again, that was a species that we sort of marked as ones with the potential to cause disease. But if you think about nuts, so what is our other nut projects? And so this is then the first year that Dee started working, and so master's project. But we need to recap and think of some of our previous surveys. So yes, of course, we detected diapathy, so we know that Phomopsis husk rot is important. What we obviously and clearly observe is that there's differences in variation between growing regions and between seasons, and that Colototricum species is also important, so we cannot ignore anthracnose husk rot. But something that for us initially was a little strange is then the dominance of Colonectria species from a single orchard in the Lavubu area. And so we picked up and we sampled Colonectria both in 2017 and again in 2019. So how relevant will this be for the entire industry? What will the impact be of a Colonectria causing disease on nuts? We don't know, but perhaps we need to take a closer look. And what I'm just pointing out here in the slide is that both in 2019 as well as in 2020 did we see reports of Colonectria causing disease of nuts from 2 different regions in China. And so as much as it's important for us to understand the species, understand which causal agent, we also need to really focus and look at biology. And so, in a sense, talking about disease epidemiology. And so this is the work that was done by Zanelli, and the project was actually sponsored by Westphalia. So for 2 consecutive seasons, Zanelli actually looked at the correlation between husk rot, so the percentage or the incidence of husk rot and macadamia nut borer damage. And she also looked at the number of stick tites in the tree versus husk rot. And so what we could clearly see is that there's a very strong positive correlation with nut borer damage. And so yes, insect damage will increase the incidence of husk rot, but we didn't see a correlation between husk rot and stick tites. What Zanelli also did was a trial that allowed us to determine which of the specific nut physiological stages are more susceptible to natural infection. So in the following slide, I really try to graphically display or explain our slides. Number 1 and 6, in a sense, represents our controls, where either none of the stages were sprayed or all 6 of these different nut physiological developmental stages were sprayed. And then so the different repeats, we focused on a specific stage where we left it unsprayed, and so we allowed natural infection to occur. And what we could see is clearly see an increase in husk rot. And this allowed us to determine that the earlier stages of nut development are in fact more susceptible to natural infection. So perhaps not only, but an early spray would be recommended if you have a history of the disease. So if we then move on and think in terms of some of our disease or pest work, first of all, the work of Ashley, and she works on Lepidoptera species. So yes, we know of at least 4 species that is causing damage, but from a survey, and we looked at 1,000 larvae, 2 consecutive seasons, by far the most dominant species that we obtained was that of Macadamia nut borer, so Tomatotibia batrachopa. But we know that species composition can change over time. So our result is slightly different to a study that was done a number of years in Levubu. And so we actually encourage growers to make sure of the species identification of the larvae in your orchard. So for this, we made use of a DNA approach. And the value of that, it also allowed us to take a first look at what the population is. So why is it important to understand populations, specifically if we think about different growing areas? And so there's a lot known that different or genetically distinct populations can actually respond very differently to biological control. And specifically in Lepidoptera, we also sometimes see small variation in pheromone blends between populations of the same species. So the following is a haplotype, a genetic haplotype network that's here constructed. First of all, I color-coded a larvae that was obtained from our 3 main growing areas, Mpumalanga, KwaZulu-Natal, and Limpopo. The size of the circle will be then the number of individuals that had that exact same DNA profile. And perhaps all that I want you to see from this is we did not see this clear separation of geographically distinct populations. And so in terms of management, I think this is good news for growers, and that we expect that the biological control should work equally effective in different regions. So finally, some updates on our stink bug work. So first of all, the project of Byron, and here specifically, he's documenting the diversity of species. So yes, a lot of information is known, and not only, but we need to acknowledge all the work that was done by Skalk Skuman to spot the most dominant. But if we really look at all the work, we can clearly see that species composition is changing and that there's more and more species that is becoming also dominant. But Byron's specific project was to actually help to construct a DNA database for us. Why do we see this as important and valuable? So we can easily identify a species by making use of a nymph. We can actually extract DNA from an egg and we can determine the species. And this is all without the knowledge and without having to rear them to an adult. Morphology can also sometimes be a little confusing, and that is because of the fact that the species can undergo color variation. But finally, of course, it's not just diversity because it's interesting, it's understanding diversity and linking that specifically to damage. So I want to share one single slide And that would then be our 2019-2020 KwaZulu-Natal survey. So note, this is not exact numbers. We received stink bugs via scouting. And so perhaps the first thought is, yes, this is really familiar, two-spot most dominant, a number of species that we already know. But I would like to draw your attention to what is now just currently an unknown stink bug species. The work is done in collaboration with Michael Stiller, so we will have a species identification soon. So important of the species that it was the second most dominant. It was present in all the farms that was part of the survey throughout the season, and it has not been detected in either Mpumalanga or in Limpopo. So now we think in terms of this species, and we need to ask what is its potential to cause damage, and this really requires further investigation. So if we think about perhaps a species dominant being in a specific area, we think about the natural environment, we think about alternative hosts. So excited to share some of the work that Gerda Fourie, our postdoc, is doing. And so she developed a detection assay that will allow us to identify alternative host species. So specifically, we started with a lab-reared isolate. From there, she extracted the gut. Then after we extracted DNA, but we specifically focus on amplifying barcoding genes that are plant specific. So this is currently our proof of concept. We were able to successfully do this on stinkbug that was reared on maize. We were also able to successfully do this on stinkbug reared on macadamia. What is currently being done is sort of our feeding trial exclusion study. So we need to know what is the number of days that we will still be able to detect the species that it came from. So our final project, of course, that is the work of Elisa Paul. And when she joined about 2 years ago, first important step for us was to establish a lab-reared population. It was very easy for Arista to really work with this stink box because we now have it at our institute. And of course, this also allowed her to really focus on one of the key aspects of her PhD, and that is to sort of look at pheromones and its potential for control. Currently, our focus is on the alarm pheromone. So what she's done thus far is she done some gland extractions. She's in the process of doing headspace analysis. She's done her GCMS. So in other words, she started identifying the components, and we're now at the behavioral activity study. So of course, we need to be able to see that there's a antennal response. And so she's optimized the system. And we know that when we do a puff experiments with a plant volatile, we see a response. Also during the course of this year, FABI really invested in upgrading our behavioral and insect behavioral room. So excited to some of that work really going on. So I'm just showing some of the components that she analyzed. So that would be 5 of the main components. And it's important that we don't see a difference between male and female. So we are, in fact, busy with the characterization of alarm pheromone and not a sex pheromone. So how to summarize all the research? I want to just say quick and accurate identification of pests and pathogens is important. Dry flower. What is the calanectra and is it going to be important? What about the polyphagous shuttle borer? Understanding variation, understanding genetic variation, because genetic variation can influence the success of specifically biological control. Some knowledge that was gained regarding biology and epidemiology. Think of nut borer damage and its effect on husk rot. Understanding that an early spray may be important. Excited about our detection assay that we've now developed. Looking forward to really testing these fly-ins, and for that we would seek collaboration from industry. And then, of course, progress towards the characterization of pheromones. I have a number of acknowledgements. So the work is done in collaboration with many academics at FABI. Thinking of Andre and Femi, the time that we spend in Australia, but they will also work with us in future with some of our disease projects. Jeremy Allison, of course, involved in all of our pheromone work. Acknowledging Elsie that is helping with some of the stink bug work. Michael Stiller Morphological identifications is really, really important. But then perhaps the most important would be our farmers that has taken part and that has really helped us to collect larvae, to collect stink bugs. But a special acknowledgement to Danrock and Mattison's farm. Danrock firstly sponsored the trees that we used, and then Mattison's farm allowed us to sample the branches that was used in the Fusarium trial. And so with that, thank you very much. Oh, so I see a question. Is the disease clinic running at full capacity again? Yes, we hope so. We still have a limit on how many students or people are allowed at the institute. So processing is still a little bit slow, but yes, we are open and running and happy to take in samples. So there's another question on how are we going to make sure that the alternative hosts, that we can pick it up. So important there is we need to have a timeline. So this is going to be difficult, but I'm really positive that there's a number of growers that really has their hand on what is in their orchard, and they will be able to help catch those early on arrivals. But yes, time will tell to see if we really are successful in doing this. And I think that is it. Thank you very much.